Hi guys, today we're going to review genetics and start getting into Punnett squares. So the first question I'd like to ask you guys is, what's the difference between a trait and character? For example, hair color is a character, but certain traits can actually be different. So you could have blonde hair, brown, black, or red, and those traits are actually different genes that are located on our chromosomes. And we're going to be working today on how those traits get inherited using Punnett squares. And we've talked about the prefix allo, A-L-L-O. Allo means other, and that's the prefix that we find in allele. So it's another form of a gene. So that would give us our traits. So an allele for hair color could be brown, could be blonde, could be red. And another example is flower color, height, some other things. So alleles are again carried on chromosomes, and we used when we were talking about meiosis, we used the colors as different genes. So when we practiced our crossing over worksheet, we used colors to indicate different genes on a chromosome. So now we're going to take that concept, and instead of colors, we're going to put letters on there. So big T or little t. And these would represent different alleles for a certain character. So in this example, the big T could be tall, the gene for tall, and a little t could be a gene for being short or vertically challenged. So different terms that we use in this chapter is the term dominant, which is a global phrase that means it just would overshadow another trait. And we typically represent those with capital letters. Big T, capital A, capital B. And a recessive one is when that particular gene doesn't show up if there's two, or it could actually be overshadowed when it's with a dominant trait. And if we're going to represent that on a Punnett square, we do that with lowercase letters. Little t, lowercase a, and lowercase b. The other terms that we use is genotype. That's actually what our genes are. So we actually would say here that we have two dominant genes, and so what we use in those terms is homo, meaning same, zygos, homozygote, dominant, for two big T's. We have a big T with a little t, that's a heterozygous, hetero meaning different. So heterozygous would be big T, little t. And when we have two little t's together, that would be homo, meaning same, homozygous recessive. And so it's important to actually note the difference between, I could have a homozygous here, and I have a homozygous here. They're different because this is homozygous dominant, which would mean that the person, if I were to say homozygous dominant, you would translate that into two capital T's. Homozygous recessive would be translated into two lowercase t's. And if I use the term heterozygous, you know that those two alleles would be different, capital T and a lowercase t. So genotype is when we actually talk about the genes, and phenotype is what they physically look like. So I've underlined here the P for phenotype, and P meaning physical, what the physical traits are. So if we take this homozygous dominant, we have two dominant genes. That's going to translate into someone being tall. So their genotype would be homozygous dominant, and their phenotype would be tall. Genotype of a heterozygous has one dominant and one recessive gene. That person would look tall because the recessive allele doesn't have a chance to kind of do all the talking. The capital T does. So the dominant gene overshadows the lowercase, and that person would actually look tall. Two lowercase t's, meaning homozygous recessive, that person would physically look short. So what do these mean? Two capital T's again would be homozygous, homo meaning same, homozygous dominant. Here we have heterozygous, and this would be homozygous, homo, meaning same, heterozygous recessive. So how would you describe these different samples? You can pause the program right now and actually write next to the letters, write out what you would actually call this. Not necessarily the phenotype, but how would you describe this? And I'll do the first one for you, and then you can do the rest. So capital A, lowercase a, would be heterozygous. So now pause the program and figure out what you would call the rest of these. How'd you do? So this would be heterozygous. This one's homozygous dominant. And the last one is homozygous recessive. And the capital A and capital A is homozygous dominant. So when we do a cross, we're really just fertilizing or mating or breeding different organisms together. So we use the term cross for that. And a monohybrid cross, mono meaning one, so we're going to cross one characteristic. So let's look at some examples of that. We're going to do some crosses, and for each cross, we're going to describe the cross as a homozygous dominant crossed with a heterozygous recessive. And then you're going to describe the percentage of the offspring. So here's some practice. I'll do the first one, and then I'd like you to describe the next three. The first one, homozygous dominant crossed, a little x, a homozygous recessive. Now pause the program and write down what you would call these next three crosses. Okay, now you can check your work. This would be a heterozygous crossed with a homozygous recessive. The next one, heterozygous crossed with a heterozygous. And the last one would be homozygous recessive crossed with a homozygous recessive. To fill in a Punnett square, you take the first organism and you separate out. This would be a heterozygous across the top, putting one letter here and one letter here, crossed with another heterozygous, so one letter here and one letter here. And for a monohybrid cross, you want to make a nice big square box and then put panes inside that. Each letter would go down, so this big capital B goes down, and then it goes down to the bottom. One letter goes over here, and the other letter goes across. Then you do the same with the other letters. The little b comes down, and the little b comes down. This one goes across, and then it comes over. So in this example, we cross a heterozygous with a heterozygous. Now if I was going to describe these genotypically and phenotypically, I would say there's 4 offspring. 25% genotypically is homozygous dominant. 2 of the 4, so that would be 50%, are heterozygous. 1 out of 4, or 25%, are homozygous recessive. So that's describing them genotypically. Now, if I needed to describe them phenotypically, I need to know what the letters actually mean. So we're going to use a big B for big, and a little b for baby, baby-sized. I'm just making this up. So here, this person would look big. This person would look big because the lowercase b is being overshadowed by the big B. Same thing here. So 1, 2, 3 are all big. So 75% phenotypically are big. But then I have a homozygous recessive, so here I'm actually seeing the baby size. So 25%, or 1/4 of the offspring would actually be baby sized. So that's how we describe things phenotypically and genotypically. So now what I'd like you to do is actually do these practice problems for yourself. Do sample 1, sample 2, and sample 3, and make sure in your notes that you write these samples down so I know which ones you're doing. Do the Punnett squares for each, and then for the offspring, you're going to describe your offspring genotypically and phenotypically, providing answers in percentages, just like what we just did. And here's another example for that. So good luck. I hope you guys do well with it, and I look forward to seeing your answers in class.